Let's discuss the history and creation of the confocal microscope. First, I would like to focus on the inventor and his reasoning for creating this microscope that has been groundbreaking for multiple fields of science. Dr. Marvin Minsky was born on August 9, 1927, in a small town I like to call New York City, New York. His father, Henry Minsky, was an ophthalmologist. Therefore, as Marvin Minsky describes, he grew up taking apart and reconstructing various lenses, prisms, and diaphragms in his household. Growing up around these objects helped Minsky expand his knowledge and develop a passion for mathematics and physics that led him to later invent the confocal microscope in 1961. Minsky received his bachelor's degree in mathematics from Harvard University and his doctorate from Princeton University. In his graduate studies, he focused on the connections between mathematics, physics, and biology. He is now an electrical engineering and computer science professor at Massachusetts Institute of Technology and researches the connections between various scientific fields. He uses his vast knowledge to research and develop ideas regarding artificial intelligence, cognitive psychology, and neural networks. Throughout his lifetime, Minsky has made many groundbreaking discoveries and inventions in machinery, artificial intelligence, and neurology. Some of his most remarkable inventions include the first neural network simulator in 1951, the confocal microscope in 1961, and the first head-mounted virtual display in 1963. Minsky has had a great influence in multiple scientific fields, but we will focus on his creation of the confocal microscope, which is now used to view and study various tissues and cells. In order to understand the purpose of the confocal microscope, one must first look at Minsky's motivation. As stated previously, in graduate school, Minsky studied the connection between artificial intelligence and machinery with biology and psychology. He developed theories on connectionistic learning machines that he related to the human nervous system. He saw many similarities between the machine's circuitry and the human nervous system. However, the field of neurology had very limited knowledge at this point in the 1950s. Minsky felt that he could solve problems in artificial intelligence at the moment, but he saw a more pressing need for increased knowledge in neurology. So, he decided to fill in those knowledge gaps first, which would later help him make discoveries regarding artificial intelligence. In order to learn more about nerve cells, Minsky tried to view the cells under a microscope. However, all the microscopes he used could only view one nerve cell at a time and did not have very good resolution. For his research and understanding how nerve cells interact with one another to form a larger system, he needed to find a way to view multiple cells at one time. This gap in current microscopy gave him the idea to form a new microscope. Minsky started working with his desired goal of producing his ideal microscope in mind. His ideal microscope would decrease the amount of scattered light and would not allow extra light into the field of view. This would concentrate the light on the desired specimen and increase the quality of the image. Also, each segment of the specimen would be able to be isolated and examined by measuring the specific amount of light scattered by the specimen instead of just a single cell. In creating his ideal microscope, he faced many challenges in the physics and functionality of his invention. As stated previously, he wanted to form a microscope that measured the specific amount of light associated with a single specimen. However, measuring the amount of light scattered by a particular object only resulted in more scattered light, which decreased the ability to focus on the specimen, or in his case, nerve cells. He found that it was going to be more difficult to reach his ideal microscope by diminishing every undesired ray than previously thought. His ideal microscope also involved viewing multiple nerve cells at one time. In order to do this, he would either have to move the specimen or the beam of light, which still maintained the 3D alignment of the optics. Also, he needed to find a form of bright light that he could harness. To conquer the challenge of eliminating undesired scattered light, he removed the scattered rays from the initial focal point by using a second microscope to image a pinhole aperture on a single point, which reduced the amount of light entering the objective lens without altering the high resolution or brightness. Also, he added a pinhole aperture on the same plane as the sample, which reduced the amount of light off the parts of the specimen that were currently out of focus, resulting in a microscope with a pinhole and an objective lens on each side of the specimen. These changes in the microscope allowed Minsky to manipulate refraction patterns of the pinholes to increase resolution and to view multiple nerve cells in a plane. In 1957, Minsky submitted his patent for the first confocal microscope, but it was not published until 1961. The microscope designed in his patent required a single image to be scanned at a time due to the single-point illumination. The scanned image was then viewed on a military long-persistence radar, which used a photomultiplier to measure the light output of the specimen. He also decided to move the specimen plate instead of the beam of light to maintain the optics of the system. For his strong beam of light, he used zirconium arcs because it was the most dependable source at the time. The microscope that Minsky designed was then altered with scientific advancements, especially with the invention of the laser. Many modifications have been added with technological and scientific discoveries to improve the microscope. All these advancements have been harnessed by many scientists, resulting in multiple types of confocal microscopes, including the laser scanning, disc scanning, and multiphoton laser confocal microscopes. Each type has its own significant improvements, allowing in various forms of high-resolution scanned images. One invention that improved the microscope was the invention of the laser as a high-energy light source that has been used to allow a shorter scanning time. Also, the invention of the computer and its ability to view scanned images has been used to enhance these images and view them for a longer period of time or indefinitely. Now let's take a look at how Minsky's invention works.